This essay example, as well as thousands of others, is available in text format on our website for free and without registration. Simply Google Ivy Panda Free Essays.
The mean number of germinated seeds in 7 days under various conditions of liquids. The control treatment had distilled water, and the experimental treatment had 0.0625 m and 0.125 m for each calcium chlorine 2, potassium chlorine, magnesium chlorine 2, and sodium chlorine solution. Comparison between different concentrations. The results indicated that the number of germinated seeds differed according to the concentration of solutions. The results of calcium chlorine 2 showed that the seeds placed in 0.0625 m calcium chlorine 2 germinated quickly on the 2nd day and attained the mean of about 10 germinated seeds on the 7th day, while the ones placed in 0.125 m calcium chlorine 2 delayed germinating and attained about 3 as the mean number of germinated seeds on the 7th day. The results of potassium chlorine indicated that the seeds placed in 0.125 m potassium chlorine germinated earlier on the 1st day than the ones placed in 0.0625 m that germinated later on the 2nd day. In the seeds grown in magnesium chlorine 2, a comparison of results showed that seeds in 0.0625 m solution started to germinate on the 2nd day, while ones placed in 0.125 m solution delayed and germinated on the 3rd day. The results of sodium chlorine showed that seeds in both 0.0625 m and 0.125 m solutions germinated on the 2nd day and attained approximately the same mean number of germinated seeds on the 7th day. Comparison between different solutions. Furthermore, the results showed that the number of germinated seeds varied as per the type of solution. A comparison of results reveals that seeds in potassium chlorine started to germinate on the 1st day, while seeds in calcium chlorine 2, magnesium chlorine 2, and control solutions started to germinate on the 2nd day. In contrast, seeds in sodium chlorine started to germinate on the 3rd day. Relatively, approximately the same mean number of seeds had germinated in potassium chlorine, sodium chlorine, and control solutions on the 3rd day. On the contrary, the mean number of germinated seeds was the lowest in seeds in calcium chlorine 2 solution, followed by the mean number of seeds in magnesium chlorine 2 solution. From the 4th day through to the 7th day, the mean number of germinated seeds depicted a similar trend in magnesium chlorine 2, sodium chlorine, potassium chlorine, and control solutions. Comparatively, the results revealed that seeds in potassium chlorine, magnesium chlorine 2, sodium chlorine, and control solutions had about the same mean number of germinated seeds, whereas seeds in calcium chlorine 2 had the lowest mean number of germinated seeds. Discussion. The objective of the experiment was to ascertain how different solutions influence the germination of seeds. Essentially, the nature and concentration of solutions influence osmosis, which in turn affects the rate of seed germination. The results showed that a high concentration of calcium chlorine 2 inhibits the germination of seeds, while a low concentration stimulates growth. Regarding the concentration of potassium chlorine, the results indicated that low concentration hastens the germination rate of seeds. In the aspect of magnesium chlorine 2, the results revealed that a high concentration slows down the germination rate of seeds. The results also showed that a low concentration of sodium chlorine hastens the germination rate of seeds. As osmosis is a physiological process, which explains the effect of a solution's concentration on the germination of seeds, Bojovic et al. Explain that inorganic ions in solutions reduce the osmotic potential of seeds and hinder the effective absorption of water for germination purposes. Seeds grown in solutions germinate at a slower rate or fail to germinate when compared to seeds grown in distilled water. The results demonstrated that seeds grown in the solutions of calcium chlorine 2 and magnesium chlorine 2 with high concentration had slowed germination rate or inhibited germination when compared to other solutions. These findings are consistent with the findings of a study, which established that the inhibition effect was higher in the solutions with magnesium chlorine 2 and calcium chlorine 2 when compared to solutions with sodium chlorine and potassium chlorine. The plausible explanation of the apparent observation is that divalent cations induce more osmotic stress than monovalent ions. Moreover, from the results, it is apparent that seeds grown in different solutions did grow at different rates but eventually achieved about the same number of germinated seeds. The rapid germination rate observed occurred because monovalent ions do not induce significant osmotic pressure on seeds, thus, allowing them to imbibe water fast and germinate rapidly. In saline conditions, seeds take long to germinate because the osmotic stress slows down the process of water imbibition, resulting in the delayed breaking of dormancy. Eventually, seeds manage to germinate even in the most saline environment because seeds imbibe water slowly until they get adequate amounts for breaking their dormancy. According to other studies, the presence of cations, such as magnesium 2+, sodium+, calcium 2+, and potassium+, in water reduces the germination rate of seeds because they create physiological drought and prevent or delay seeds from breaking their dormancy. The findings of this study are similar to other studies because the design of the experiment ensured that the findings were valid and reliable. Therefore, the findings support the hypothesis that the rate of germination in seeds grown in solutions is slower than the rate of germination in seeds grown in distilled water. As the experiment used 2 concentrations of a given solution and 3 replicates, the outcomes do not provide robust results. In this view, a follow-up experiment should expand the range of solutions and the number of replicates to provide a clear trend of variation in the germination. Moreover, there is a possibility that the experimental design led to biased outcomes for each student recorded own data. Hence, a blinded study is appropriate to prevent researchers from introducing their biases into findings. In conclusion, the experiment has demonstrated that a high concentration of calcium chlorine 2 and magnesium chlorine 2 inhibits germination, while a high concentration of potassium chlorine and sodium chlorine slows down the rate of germination. If you want to find more works like this report on seed germination experiment, results and discussion, head over to ivpanda.com. It has a collection of samples with thousands of submissions covering virtually all academic subjects.